Well, welcome to the Science and Faith podcast with Dr. James Tour. I'm Dr. James Tour. I'll be your host, and you can find my credentials at jmtour.com, and you can find my social media sites at drjamestour.com. I want to, as with my other interviews, if you want to be part of the audience to be able to participate in future podcasts, to be able to ask questions, you could go to signup.drjamestour.com and get on the list for that. This is a recorded interview with a live audience, and that audience is going to ask questions in this recorded video, and that's what will then go up. Now, I'm a practicing scientist, but I love Jesus so much. Jesus means everything to me. And if you want to know Jesus, if you don't know Jesus, I offer you this. You can send an email to me, tour@rice.edu, and I will meet with you via Zoom. I'll set up a time to meet one-on-one with you and share about how I came to a relationship with Jesus Christ. This is not for believers. This is for unbelievers. And the unbeliever themselves have to send me an email requesting that time. So if you have a friend or somebody that you want me to share with, that friend has to write to me, or that child of yours has to write to me requesting that time, and to hear about how I came to faith in Jesus Christ. I will gladly meet with them. It's not a burden for me. I will gladly do it. And with that backdrop, let me introduce our guest for today. It's Dr. Brian Miller, and he is he's research coordinator for the Center for Science and Culture for the Discovery Institute in Seattle, Washington. He holds a BS in physics with a minor in engineering from MIT and a Ph.D. in complex systems physics from Duke University. He has extensive expertise in the application of thermodynamics and information theory to the problem of the origin of life. His other interests include how the effects of mutations on the stability of protein folds relate to the rarity of functional proteins and their application of engineering concepts in biological systems. He is also a technical consultant for thestartup.com, a virtual incubator dedicated to bringing innovation to the marketplace. And with that, I introduce our guest, Brian, and Dr. Brian Miller. The first question I want to ask you is this. With all of your education, how is it that you can believe in something so amazing as the physical resurrection of Jesus Christ?
Well, it'd be helpful if I shared a little of my background. When I went to MIT, I became pretty convinced God didn't exist. I read Richard Dawkins' book, The Blind Watchmaker. I was told the Bible is just a bunch of fairy tales. And I realized that if God didn't exist, then life was pretty much pointless, that I'm just going to die and cease to exist. It doesn't matter if I'm happy or sad or kind or cruel. And I remember just in my room, I said, God, I don't know if you exist, but if you do exist, you have to prove it to me because I'm a scientist. I didn't know if that was fair, but that's just the way it was. And that sort of put me on a course where I could really see God's providential hand in my life, where I was able to meet leaders in the field of faith and science, people that were experts in the resurrection. And it was really a two-part journey. One is I came to realize that science points to a creator at every level, from the design of our universe, to the design of our planet, to the design of our cells, to the design of ourselves. I also came to realize that the evidence for the resurrection of Jesus is overwhelming, both from the history, from eyewitnesses, different things like that. But also, it was encountering God very personally, seeing miracles, experiencing the love of God. So it was both an intellectual journey and a personal journey that I came back to faith and realized. And I think this is really what made the difference for me, because a lot of people want to seek after truth as a hobby. But I told God if he was real and he proved it to me, I would dedicate my life to serving him, that I would give him everything. And he honored that request. And since that point, that he's shown me that he exists, that Jesus Christ is the Lord of creation, that he died for my sins on the cross, I've dedicated myself to speaking the truth about the evidence for God in Jesus Christ and about sharing his supernatural power to transform lives.
That's great. That is great. What an explanation. So it's interesting that you went through this phase where you didn't even believe anymore after reading some of these books, and then it became obvious to you through science. That's a lot of it. I came to know Jesus Christ when I was a freshman in college, and I loved him so much. I loved Jesus so much. It was never a question after that. And the science only underscored all of this, the beauty of the creation, just the beauty of it. And let me start with a first question here. It's a little bit long, but I want to set the stage for you because you are in from what I know just an excellent thermodynamicist. And so, Dr. Miller, you're an expert when it comes to explaining thermodynamics, entropy, and energy, and how it relates to living systems. The simple meaning of the second law of thermodynamics says that systems move from lower to higher states of entropy. And so it follows that the spontaneous formation of a living system by chance is unlikely. As Hoyle put it, it's like the chance of a tornado going through a junkyard and assembling a Boeing 747. But some systems do move from high to low entropy state. For example, ice has a lower energy state than water. Can you explain to the average listener the difference between what happens with ice and the requirement for life, and how all that relates to pre-biology, life's building blocks, and the requirement for life in the first place, where abiogenesis is before biology takes over? This is the origin of life. How did that first living system form?
Sure. And entropy is simply a measure of the number of ways that a system can exist in a certain state. So people say that, well, water has more entropy than ice because there's more configurations of molecules that would correspond to water than to ice. So to say that systems tend toward from low entropy to high entropy is simply saying that systems tend to go to states that are more probable. There's more ways they can happen. It's as simple as that. So if you imagine rolling a dice, there are very, very few ways that you'll roll 1,000 sixes in a row. I mean, there's only one, in fact, and there's a lot of ways you could roll other numbers. So people have argued that the origin of life can't happen because it's too improbable to have a lot of atoms in a state that corresponds to life. That argument is incomplete because there's also another driving tendency of nature. Yes, nature will typically go from order to disorder or from low entropy to high entropy, but nature also wants to go from high energy to low energy. And those two tendencies at times can work against each other. So the reason that water will freeze and turn to ice in low temperature is because the attractive forces between the water molecules mean that ice is a lower energy state than water is. Now, what happens is when water becomes ice, it releases heat into the environment. So when that heat goes into the environment, the entropy of the environment goes up by a larger amount than the entropy of the water goes down. So to think of it in terms of loaded dice, the probability or the entropy corresponds to how hard it is to roll a certain set of numbers on a dice. But the energy relates to how much the dice are loaded. So in nature, the dice are loaded for low energy. But here's the problem. Life is a state of both low entropy or very high order. It's very improbable. And it's a state of very high energy. So for simple chemicals on the early Earth to become a cell, they both have to go to low entropy, a very, very improbable state, and they have to absorb energy from the environment to increase what's called their free energy. That's a physical impossibility that never, ever happens in the universe, except if it has help, which I'll get into.
Okay. With that, I'm going to turn this over to you to show us some slides, and then we're going to get back with a series of questions.
Okay, certainly. So what happens is if you look at the argument and how it's evolved over the last few decades, the issue that people have thought about is what if we're in a system that's driven by energy? So it's driven outside of equilibrium. And what happens in these systems driven out of equilibrium, you get what's called self-organization. And that's kind of like if you have a tornado, the flow of energy and mass into the system creates a funnel cloud. It self-organizes. If you boil water, you get roll patterns. That's a self-organizational pattern. And people have said maybe that's how life formed. Maybe if you shove energy into a system, that will create a self-organizational process that would create a cell. And that's actually completely false. Because while the order in these systems driven from equilibrium takes place at the macro state, at the large level, you see order. At the micro state, at the molecular state, you have more disorder. You're actually generating entropy. And that gets into the work of people like Jeremy England, that gets into what are called the fluctuation theorems. And the last few decades, there's very sophisticated theorems related to non-equilibrium thermodynamics, like the Evans-Searle fluctuation theorem or Crookes fluctuation theorem. And what they've shown is that it's as improbable for a chemical system to go to both low entropy and high energy away from equilibrium as it is near equilibrium. So that means arguing that any physical process could take a bunch of chemicals and form a cell is the modern day version of a perpetual motion machine. Now, the only way that life does it is it has engines. It has these molecular engines like ATP synthase. And what these engines do is they take energy from the environment. It could be things like glucose, it could be sunlight, it could be some other chemical. And it converts that energy into a form useful for the cell, like ATP. So engines are necessary to provide the energy required for life. Also, cells have information. What cells have are enzymes and other structures, and enzymes are proteins, and they're a chain of amino acids, and there's 20 amino acids that form a protein in the same way you've got 26 letters in the English alphabet that form sentences. So you can think of these protein chains as like sentences where the alphabet is 20 amino acids. And if the information in that chain is right, the right sequence, it folds into the right three-dimensional shape, and that enzyme can then drive the right chemical reaction. And that enzyme can also extract energy from these high energy molecules like ATP and use that energy to drive the reaction in a way that it would not normally would go. So it can actually become a higher free energy product. So it's information and engines that are necessary for life. And I had a wonderful exchange with Jeremy England, and I made that point. And in his response, he either explicitly or implicitly confirmed all of those claims. Now I'm going to go to my slide presentation. And this is a very recent paper. It's 2018, and one of the authors is Michael Russell, and he was one of the architects of like the thermal vent theory of the origin of life. And in this paper, he actually acknowledges all of these points I just made, because he says for life, you've got to have engines that provide energy in a form that can power cellular processes. So you have to have these engines, and you have to have machinery that selects specific chemical reactions and creates the right small environments to produce target products. And it also blocks other reactions. And again, this gets back to information, because these machines he's referring to, again, are enzymes that have the information in the sequences. And he talks about how in life you have to control the reactions based on cellular and external conditions. So if it's hotter or colder, or if the pH is different, the cell has to sense the external environment, sense its internal environment, and control reactions in that fashion. And there's also another research paper that was by, that talks about the challenge of those control systems. So what I'm showing you now is simply an example of a feedback loop in a cell called the lac operon. It's one of the simplest feedback loops. And this is necessary because this particular operon, which is a set of genes, controls the digestion of a sugar called lactose. And what happens is when there's lactose in the environment, there's a feedback that causes the enzymes to be produced to digest the lactose. But if there isn't lactose, then the thing shuts off. So it's this feedback loop. And you have to have these sort of control systems throughout life. And this paper, it talks about the difficulty of that. And what it says, noteworthy, the probability of instability of a randomly selected state increases rapidly with increasing size of the system. Its stability and thus its existence over a prolonged period of time depends crucially on the numerical values of kinetic parameters and regulatory interactions. In other words, if you imagine the origin of life and just through some miracle, a cell almost forms, it has all of the proteins, the DNA, the cell membrane, but the feedback loops are not quite right. They're either too strong or too weak, or the metabolites, the chemical products aren't in the right proportions. The metabolism shuts down and it quickly will destabilize and decompose into simple chemicals. So this is really, really challenging. So what happens is you have other researchers like Yasser Demirel who talk about the same issue. And here's a great point. It says the ATP synthesis in turn is matched and synchronized to cellular ATP utilization processes, since energy and matter flows must be directed by the information for them to be functional and serve a purpose. For living systems, the information is the cause and the entropy reduction is the result. Energy plus matter plus information is a locally reduced entropy. So what all these authors are saying is for a cell to function, you again have to have energy provided, and you have to have machinery that directs that energy in such a way that allows a cell to function. And you have to have highly specialized control systems to control these chemical processes, or else the whole system destabilizes, and that all goes back to more information in DNA. So this is what this author and other authors are saying. Now, here's the really key point. How does he deal with this? And this is the second part of that same article. It's part 2. And here's a quote from it. And what he says is, he says, we claim in particular that it is untenable to hold that life-relevant biochemistry could have emerged in the chemical chaos produced by mass action chemistry and chemically non-specifically energy inputs, and only later have evolved its dauntingly specific mechanisms as a part of evolving the rest of life's features. So what this author is saying, who is really very respected in the origin of life community, is that it is a physical impossibility for chemicals on the early Earth and random energy inputs to produce a cell. Now that's shocking, but it's interesting to look at how he responds to this challenge. And this is really how all origin of life people deal with this problem. Instead, we claim it had to have been launched simple and specific, and thereafter have been forced by the scythe of natural selection to maintain the necessary specificity standard at each evolutionary increment of complexity. More specifically, all the devices of life, metabolic and structural, must have been invented by it, that's natural selection in situ, step by step, and while in flight, starting from the simplest possible inputs. And learning itself by trial and error which small changes themselves occurring by chance were useful. Each incremental step in the evolution by creeps complexity building process necessarily being vetted by the system both for fitting in and contributing more utility than cost. Now, I think that's really striking, because what you're seeing in this case is you have a situation where origin of life experts freely acknowledge that a cell is too improbable to come together by chance. They acknowledge that in order to have this incredible chemistry, you've got to have machinery that directs the chemistry. They acknowledge that you've got to have control systems. And they acknowledge that there's no physical process that can deal with this. So what they do is they appeal to natural selection. The problem is you can't have natural selection if nothing is replicating, if nothing is reproducing. It doesn't make any sense. He's talking about this sort of chemical soup in a thermal vent, and there's nothing replicating, so natural selection isn't real. So origin of life people appeal to natural selection, not as a real process, but kind of as a demigod, almost like Plato's demiurge, where it has the intelligence to create a cell. It has the intelligence to select the right chemicals. So they're invoking intelligent design without acknowledging it through the guise of natural selection. And I'll just conclude this section by mentioning they do at times appeal to self-replicating molecules like self-replicating RNA. And the idea again is you've got RNA that's replicating, it's reproducing itself, it goes through small changes, small mutations, and then natural selection selects what's called an abzyme, which is simply an RNA molecule that acts like an enzyme. But here's the problem. If you look at all research on self-replicating molecules, like Spiegelman's monster is a classic example, What you find is replication of molecules only happens if you've got the molecular machinery borrowed from cells to make it happen. If you were to take a bunch of RNA, pour it into a thermal vent or into a small pool, the RNA breaks apart. It won't replicate without help. But even if it did, here's the real problem. What happens when you have self-replicating molecules, RNA or anything else? They become, the ones that dominate are the ones that are more efficient at replication. And they typically become smaller and smaller and more streamlined until all they do is self-replicate and nothing else. So again, all of this appeal to natural selection makes absolutely no sense. So let me stop there and we can go back to the conversation.
Well, this is interesting. You know, we had a project a number of years ago. It was funded by the National Science Foundation to build self-replicating molecules, but we could not use nature's constructs. So we said, how hard would it be to have a parent system, have monomers come up and interact with those, and then hook those monomers together? So now we've got a duplex, and then split that apart and have two new mother complexes that could do this. And not using nature's constructs, but having to think of it using our own minds. And what killed us was the fidelity. So our yield might be 70%. So you've got 30% of other things in there. And now you try to do another replication cycle, your yield from the parent is 70%, and that 30% is making other mismatches. And so within a couple of cycles, it was just an utter mess, an utter mess. And what people will do, they'll appeal to selection to purify that mess. But as chemists, you don't have selection. Selection is something that comes with biology, but this is pre-biology. There are no selectors. It's up to the chemist to figure out how to pull those things apart. And it's very difficult because when you're missing one unit from a polymer, it's very hard to distinguish those and pull them apart. So it was just an utter mess. And after 3 years, we never continued with the project because I thought, this is crazy hard.
Yeah, and you've hit on such an incredible insight, is that if you look at all plausible prebiotic situations, what happens, and you could go to the Stanley Miller experiment, you could do whatever, is you always have this intractable mixture of different compounds. And it's very unlikely that only the right ones are going to link together. And you know this but so much better than I do. But even if you had the help of enzymes, like a polymerase, what you run into is Eigen's paradox, that if you have, let's say, a self-replicating RNA or whatever, and if on average you have a lot more than one change every generation, you're going to lose all your information. You're dead in the water. So that's the big problem, because the way cells deal with this problem is they've got error correcting mechanisms. Now, the problem is, in any origin of life scenario, is before the cell has enough information to provide those error correcting mechanisms, it needs those error correcting mechanisms. So Eigen's paradox kills self-replication long before a cell is large enough to have the error correcting mechanisms to correct the errors to a level of high fidelity. So you hit the problem nail on the head.
And this is what the chemist is faced with. We who do chemistry, who build molecules for a living, hear these origin of life arguments, and they make no sense to us because they say selectors, but generally the selector is more complex than the thing you're trying to make. How did you make the selector? And what gave that selector the ability to select out what you wanted to do? Without having these error correction in biology, biology dies very, very quickly because there are errors that take place. Even though in biological systems, the yields can be quite high, without having enzymes that fix it, it's just, this doesn't happen. You don't have those yet. You don't have those in origin of life. So it's a very, very hard argument. What I see is that that people throw out very simplistic ideas and they say selection takes care of it. You know, most researchers who actually do synthesis within Origin of Life, they don't willy-nilly throw out that term because they know people are going to call them on it. Where is the selector? But what's done is a very tricky little thing, is they'll do a reaction and they'll get, say, 10 different products. And the difference between what they want and some of those products is very, very small. And they separate, and each one of these products is a peak. So they'll get 10 peaks in a very sensitive instrument called an HPLC. And they'll say, see, it is there. It's there. Now they go on to the next step, but they don't use the little blip that's there because it's too hard to separate. They'll buy that little blip or they'll make it using a natural method, and then they'll carry it on because they know they can't take that impure material and carry it on. Yet they throw that at the doorstep of nature, saying somehow nature figured out how to separate this thing. It must have been a crystallization or something. Well, if it's that easy, why don't you do it? And it's not just one step, it's step after step after step, such that the chemist becomes the selector. The chemist has said, that's the one I want. And then they've gone and they haven't even taken that out. They've bought that. They've bought that entity. And then they've carried it on in the next step, get 10 more things. They see the one that they want. They said, see, we've done it. And then they buy the next one because that is an utter mess. Chemistry is a mess. And wouldn't you say it's hard to do chemistry when you can't remove the impurities?
Oh yeah, no, absolutely. And here's what's really interesting is if you go back to the thesis that for cells to function and for biochemistry to move forward, you have to have engines, machinery that produces energy, and information to direct that energy in a way that's useful, the energy and the mass. Guess what happens in all these experiments? It affirms that thesis, because what happens is when you take a chemical and you make it highly concentrated and very pure, you're increasing its free energy. So while a cell has like ATP synthase, you've got a chemical factory that puts in the energy needed to produce this highly pure and purified chemical. Also, if you look at the sophisticated chemical protocols, the lab protocols, where they change conditions, they change reactants, they change pH, and et cetera, the information in the instructions is what constrains the outcome. So information will always, if it's used properly, constrain outcomes. It selects certain outcomes and remove others. In the case of an enzyme, it's the information in the sequence of the amino acids. In the case of a laboratory experiment, it's the information in the lab protocols that sets the conditions to get the results you want. So all these origin of life research, no matter what you're doing, demonstrates the need for engines or energy converters to supply energy, such as concentrating a chemical, and information to direct your outcomes.
That's exactly right. I mean, there's a huge amount of chemical input from the investigator himself or herself doing the very reaction. So to even think that this could happen without this input is unrealistic, because what I do is I go back and I read the experimental protocols and I read the supplemental experimental protocols. And it is filled with adjustments of pH, adjustments of concentrations, and you have concentrations and adjustments, and you shine a light at this time for this many minutes, then you turn off the light, and every one of those is information being input into the system.
Mm-hmm. Exactly. And
Right.
even, I showed you that one paper at even a deep theoretical level, the most savvy origin of life researchers are realizing that information is something beyond mass and energy. Like a great examples are Sarah Walker and Paul Davies, who are just great researchers. And they talk about in their paper, The Hard Problem of Life, that physics and chemistry will not produce a cell. That we have to think beyond that, and we have to talk about, and they talk about the causal power of information, is if information existed and it has a certain power over matter and energy. And of course, from my theological background, I would say that's an intelligent designer. That's God at work in the world, producing empirical results. So even though they're not religious and they won't go there, they still recognize that information is something beyond matter and energy, which is needed for life to work.
So take that for me a little bit further. What is the positive the positive evidence for design in biology? What's the positive evidence for
Yes,
design?
yes. Now, first of all, let's do a thought experiment to really appreciate the answer to that question. Because one of the challenges people face when they address this issue of design in biology is they ask, is there a scientific method to detect design? And the question should be looked at a little bit differently, because the study of biology or the origin of life, either way you want to look at it, is really under the field of engineering, because you're dealing with an engineered artifact produced by an intelligence far, far greater than ourself. So biology is essentially reverse engineering a technology far beyond ourself. So let's do a thought experiment. Imagine that you find a spaceship somewhere in Area 51. And two groups of scientists come together, and one, for whatever reason, refuses to believe in aliens. For whatever reason, they just philosophically can't consider that possibility. So they assume that the spaceship is simply a product of natural processes. While another group believes in the possibility of aliens, and they think, we think the spaceship was engineered by an intelligence. How would they go about studying that spaceship differently, do you think?
Well, they have two very different starting points.
Right. And how does it affect the way they look at the spaceship, the sort of questions they'd ask, the sort of ways they would probe it?
But one person has to only appeal to a natural process, and the
Right.
other person can look at this from outside that and say there are other processes that may be acting that we don't know about.
Yeah, very good. So what happened is the first group that didn't believe in aliens would say, well, this was probably produced by natural processes. They would look at maybe volcanoes would produce molten metal that would flow into rock crevices to produce spaceship parts. And then maybe tornadoes or earthquakes would cause those parts to come together and assemble in the right way to produce a spaceship. How far do you think they would get with that research project?
It'd be kind of hard to ever show that to work.
Yeah, it's kind of like Origin of Life. Now, the second group that believes in design would say, we think this is engineered. So we're going to look for the purpose behind the parts. We're going to look for design patterns in this spaceship that match design patterns that we actually do. So if you look at the people that make progress in biology, it's the same thing. If you look at all this Origin of Life research, it's pretty much not gone any places you talked about. But what's happened is there's been this shift in the study of biology, particularly in the field of what's called systems biology. Because what's happened before is if you assume life is simply the product of blind chance and time and natural processes and natural selection and horizontal gene transfer or whatever, then there's certain assumptions you have about life. One is you'll assume that nothing in life has a real purpose. Any appearance of purpose is an illusion. It's just sort of an artifact of natural selection. They would typically assume that what you see is suboptimal in design. It may work, but it's probably not as good as it could be if it was designed by an intelligence. If you look up something like the argument from poor design, if you read Stephen Jay Gould's description of the panda's thumb or Nathan Lentz's book about human errors, you get that idea. They talk about this, how evolution predicts suboptimal, clumsy, poor design. They'd also predict it would have very little resemblance to human engineering. Now, why is that? Because if you envision that a blind, undirected search, perhaps with natural selection, could stumble across some solution to like locomotion or energy production, you have to assume in this vast search space, there's lots and lots and lots of ways for you to solve these problems if you're going to stumble across any of them. So you would never expect to randomly nature to stumble across something that looks like human engineering. That would be extremely rare. What's happened is in systems biology, people have found that all of the assumptions of a scientific materialism, which is the idea that only matter and energy exist, everything's a product of natural processes, the assumptions of evolution have proven to be false. And what they've had to do to advance in biology is replace them with design-based assumptions, language, and tools. and methods. And for that, I'm going to go back to, I'm going to go share my PowerPoint slide. And here, just to kind of make my point, this is a chapter in a book by David Hankey. He says, biology is sick. Fundamentally unscientific modes of thought are increasingly accepted and dominate the way the subject is explained to the next generation. The heart of the problem is that we persist in making literal sense of the world that we know to be senseless. And the whole point of his article, the teleology, the explanation that bedevils biology, and teleology just means design, is David Hankey's complaining that biologists talk about artifacts in life as if they were designed. He says that's wrong. Thinking that something's designed in life will lead you in the wrong direction. That's the whole point of his essay. But this book is really a wonderful book. It's System Modeling in Cellular Biology. It was put out by MIT. This was out 15 years ago, and I really think this was the start of this great revolution in systems biology. And here's a great quote from the book. A hope for understanding complexity in biology, then organized, is to uncover operational principles through a, quote, calculus of purpose, by asking teleological questions such as why cellular networks are observed given their known or assumed function. So again, scientific materialism says there shouldn't be a purpose, and that's misleading. But we now know that if you look at the purpose of biological structures, that's what's going to lead you to their understanding. And again, I talked about how evolutionary assumes that things are suboptimal, but let me read you this. From engineering, it is known that feedback control plus feed-forward control enabled by fast and, if possible, remote advanced warning sensing is the most powerful mechanism for providing robustness to fluctuations in the environment and the component parts. The heat shock response in E. coli appears to employ exactly the same principles. So what you see consistently is what people used to think was poor design, like the panda's thumb. As science advances, they see as optimal design, as extraordinary design. So what's happening is if you look at researchers like Balick from Princeton, they're realizing if they assume that life is optimally designed, that leads to correct predictions. And what they're finding is that many life processes, like photoreceptors in eyes, operate at what is close to the theoretical highest efficiency that's possible by the biology. Here's another example. And this is really a great point because people will say that biology is different from human engineering in many ways, and it is. But let's see why that's significant. We adopted a high-level view of cellular systems by combining biology and engineering approaches. This perspective does not want to disguise large differences between the two types of systems. In fact, biology often shows a more remarkable design than technology. And then here's another example. An often noted reservation against this type of analogies between biological and engineered systems we brought forward states that these two types of complex systems arise in fundamentally different ways, namely through evolution versus purpose-driven top-down design. So an evolutionary assumption is that you're not going to see purpose-driven top-down design. You'll have things that come together by chance, and maybe they'll form something useful coincidentally. But what has happened is these engineers who've looked at biology have realized that things in life look like they have a purpose, and it looks like top-down design, or that purpose led to the need for, let's say, components that have to be fit together perfectly to achieve that purpose, and all the components are needed. And then those components have subcomponents that have to be perfectly fit together to create the component, which then leads to the final product. So again, this is the exact opposite of what an evolutionary process should produce, is this top-down, high-efficient design that looks like human engineering. And this kind of leads into a beautiful set of lectures that Paul Nelson, one of my colleagues, has produced. And this will be available tomorrow. So if anyone viewing this looks up design triangulation as a path for biological inquiry, it's on Evolution News, and Paul Nelson, you'll find this set of slides that he's using. And what he's talked about is how you can have a design-enabled biological research program using this top-down assumption. And the basic idea is that when we discover a complex system performing specialized functions, we assume that a rational logic, a well-matched parts are enabling that functions. So in other words, when biologists look at some system they discover in life, and they assume that it has a purpose, they can recognize that that purpose requires certain components. And if some of the components haven't been found, they can predict that those are the components that will be found based on the purpose for which the system was created. Paul describes this as design triangulation. You have some black box, which is like a biological entity. You notice that there may be two components of a system that have been identified. You know, for that system to work for its intended purpose, you have to have another component. And then the research is to look for that component. Now, Paul has a lot of examples, but I just want to talk about one particularly insightful one. And that's a discovery of the genetic code. Because Watson and Crick recognize that DNA has a structure and that it includes encoded information for proteins. And that would mean the amino acid sequence we talked about. And DNA has 4 letters, A, C, T, and G. And sets of 3 letters together form what's called a codon. And there are specific amino acids attached to specific codons. In some cases, it's 1 codon for 1 amino acid. Sometimes it can be much higher. And what happened is they recognized that if you look at the chemical nature of these nucleotides and of the amino acids, they don't really fit together because DNA carries information, but is chemically non-specific. You just have these hydrogen bonds, but amino acids have specific geometries which require a system to recognize them. So the question becomes, how do you go from DNA, these 3 letter codons, to a particular amino acid? It can't happen through direct templating because they're not chemically connected. So what happened is Crick, because he decoded messages in World War II, is he proposed that there was an adapter molecule. And there were 20 adapters at least, and each adapter molecule would connect the codons with a specific amino acid. He also predicted that there were specific enzymes that would link these amino acids to these adapter molecules. And this was very much this design triangulation logic, looking at the top-down purpose of these decoding processes. And of course, people eventually found tRNA, 20 tRNAs that relate to the different amino acids. And they have found the amino acyl enzymes, which linked the tRNAs to the amino acids exactly like Crick predicted. And what's really interesting is Watson did not like this adapter idea. He says, I do not like the idea at all. More to the point, the adapter mechanism seems to me too complicated to ever have evolved with the origin of life. And that's a key point, because for this system to work, you've got to have a adapter, a tRNA, for every single amino acid. You also must have the enzymes which connect the amino acids to the tRNAs, and that all has to happen at once. If only one piece were missing, the translation would not work. You also, at the same time, have to have the protein sequences encoded into the DNA, and you have to have the machinery that will actually perform the translation process, that will start linking together amino acids based on the DNA code. And of course, that's a ribosome. So what happened, it was Crick's design intuition that led to an understanding of biology, but Watson, because he was trapped in the framework of scientific materialism, he did not believe in design. It was difficult for him to believe because the difficulty of it coming together. Now let's apply that to the origin of life. Because if you look at the origin of life from an intelligent design perspective, you recognize that life requires a lot of different pieces. It requires structure, metabolism, et cetera, et cetera. Now, here's what's super interesting, and this is the part that really excites me, is you have scientists or engineers or mathematicians like von Neumann that have asked the question, what is the simplest possible self-replicating machine you could produce? And people have applied this to robots. They've looked at this with, like, NASA has looked at what does it take to have a self-replicating space probe. And at the same time, what happened is biologists have been looking at what is the simplest possible cell that could exist. And for a cell simply to maintain itself out of equilibrium, the smallest you get is maybe 100 amino acids. And that's an incredibly optimistic list. It's almost certainly many times greater than that. But they've looked at the minimally complex self-replicating cell through a lot of various means. And what happens is when you look at the engineering analysis of the simplest components of self-replication, That design perspective allows you to predict an incredibly large number of components that must be in a cell. And what happens is a cell based both on the engineering analysis and on studies of the minimally complex genome means that a simplest possible cell has to have energy production, selective gateways that allow certain things in and certain things out, active transport to force things in against the natural thermodynamic tendencies, information processing, manufacturing, auto assembly, automated repair, sensors, signal processing with algorithmic control, actuators that carry out instructions, control systems, and global coordination. This must be in anything that even resembles a cell, because if you don't have all these components, the cell breaks down and decomposes back into simple chemicals. So an intelligent design framework allows you to predict the components that you'll find in a cell. Now, even beyond that, because engineers have enormous experience with things like negative feedback loops, control systems, they can predict in advance what will be in a cell, not the details, but the functional components. Now, here's a wonderful paper. It's called A Survey of Engineering Models for Systems Biology. And what they did is they came to this conclusion. It says, as a discipline, systems biology shares many characteristics with engineering. In this paper, we identify the cell as an embedded computing system, and as such demonstrate that systems biology shares many aspects in common with computer systems engineering, electrical engineering, and chemical engineering. And what they do is they go through and they say, we know that what they describe as embedded computing system needs digital logic. Here's what those components look like in a computer, and we can identify the components in a cell which carry out the function of digital logic. You need a microarchitecture, as you see in a cell. You need information system architecture with several subcomponents. We can find and predict what those components will be in a cell, not the details, but what they're doing. And then you can see what the actual chemistry is that does it. And you need an operating system. So again, what happens is when you look at life from a design framework, you can actually make progress because you can ask the question, what do these systems do? And from an engineering perspective, you can anticipate what those systems must contain in terms of their functional relationships. And then you can go look for those structures that perform those functions. And very often that leads to productive research. Again, if you believe in a blind, undirected process, you assume that it's going to be clumsy, suboptimal. If you believe in design, you believe it's going to be optimal, and time again, that prediction is proven true. Now, that's just, touching the surface, but that's maybe a good start for the conversation.
That's just terrific. That is just terrific. I mean, you started out talking about the terms that are used, and they're all biological in origin. It just always mystified me as a synthetic chemist. We never spoke of molecular evolution. We never spoke of selection. I mean, we just had to make molecules. This has been co-opted from biology and brought in, and so it speaks of now you're introducing life into non-life to try to get it to go. And then, what I've always found fascinating, many people don't follow me on this, maybe you will, is that when we build robots, when people build robots, you never see us using biology to build a robot. We use silicon, we use composites, we use aluminum, we use wires, we use printed circuit boards. We don't use biology. Why don't we? Because we don't understand how to get this stuff to work together. And we don't use biological components to make our robots, although if we could, they'd look a lot more human-like, they'd look a lot more biology-like, but we don't, because it's so hard for us to understand those systems. And that's what we're up against. I'm gonna have to open it up to the audience now. We're gonna start taking some questions because
Sure.
You and I have had a great time together, but we've got to give these people a chance. In Origin of Life, people ask, what are the requirements for creating the first living cell? But can we properly answer that question, assuming that the first living cell occurred millions or even billions of years ago? Wouldn't a cell back then be much simpler than what we consider a simple cell today? What biochemical and informational requirements, if any, would be different in the Origin of Life inquiry for the first cell versus what we know of cells today? And before you answer that, let me just say, you said we need minimally 100 amino acids. I think you meant genes. We're going to need minimally 100.
Yes, I'm sorry. Amino acids, you're correct.
Yeah,
Proteins.
we're going to need minimally 100 genes, and I had heard, we're going to need minimally 256. We need some number of genes. We need something, but what would be the minimal cell? I think you covered a little bit of that, but answer it again.
Sure. Right. And when I referred to the 100 genes or 100 proteins, which they code for, that wasn't a cell. That was simply, this is a great question. That was simply the machinery needed to produce energy that would keep a cell away out of in thermodynamic equilibrium, disequilibrium, or we call it homeostasis. So if you start with chemicals, what happens is as you move that system away from equilibrium, the further you're away from equilibrium, the more the concentrations are different from the environment, the more you've got proteins that are breaking apart, the more the thermodynamic forces are trying to pull you back to equilibrium. So what happens is you've got to provide energy at every stage of the origin of life to just simply fight against the thermodynamic forces pulling you back towards equilibrium. And that's why people like Nick Lane, people like Mary Hansma, they're trying to figure out, could nature produce this energy? And if you look at what could nature produce, either in a thermal vent or mica sheets, it's the tiniest fraction of what would be needed for any but the earliest stages of the origin of life. So what happens, as soon as you go out from equilibrium, you've got to have this molecular machinery that fights against the thermodynamic forces pulling you back. It's got to repair the proteins, it's got to repair the cell membrane, it's got to keep the concentration different from the outside. So 100 genes, 100 proteins is an absolute bare minimum just to fight against the thermodynamics. Anything less than that, and any way you move away from equilibrium, you move right back. Now, if you're going to talk about self-replication, that's much harder. And I showed you from engineering, there's a theoretical limit to how simple a self-replicating system can work. You've got to have energy production. You've got to have information processing. You've got to have control gates. So you ask, well, how simple is a cell? You're going to get, if you look at work by people like Venter, you're not going to get much less than 300 amino acids at an absolute bare minimum. And that's with mycoplasma, which is a parasite. So it lives off a living organism.
You mean 300 proteins, 300 enzymes?
Yeah, 300 proteins.
You said 300 amino acids.
Yeah, sorry about that confusion. 300 genes, 300 proteins.
Genes,
and if you look at free living organisms, what happens is free living organisms are in a more unstable environment, so they need more genes to deal with that environment. They have to build everything themselves. It's a difference between how you would pack to stay at the Hilton for the weekend versus the middle of the Gobi Desert. You need a lot more equipment if you're in the wild. So the simplest cells we know of have probably have over 1,000 genes and like a megabyte of information. So if you think about how simple can a cell get before it can no longer fight against thermodynamics, it can no longer self-replicate, you're dealing with hundreds of genes at the absolute minimum. Just to fight against thermodynamics is at least 100. So to go from here to here is an enormous barrier where you need probably at least like a megabyte of information, and you need very complex energy tools. So that's a situation.
Okay, good. Let me see here. In information technology, redundant systems fall under risk management, not necessary for operation, but to protect against data loss. Data risk management isn't necessary for operation of the bits, but engineers intentionally put that in for preservation of those bits. Example, RAID, one storage array, has 2 duplicated hard drives. Where one drive can crash, the system can still operate and replace a replacement drive can be rebuilt using the good drive, all without loss. We have this same type of thing with DNA's double helix. What is the origin of the data risk management characteristics of DNA? It's a good question.
Oh, that's a wonderful question. I'm not a biologist, so I've learned enough biology to deal with what I deal with. So I would encourage you to have someone who's an expert in DNA to really flesh that out. So I'll just give a very bare bones description. Yes, you've got 2 strands of DNA, and what happens is there's error correction mechanisms when you replicate DNA. and they can tell if one of your nucleotides isn't matching the opposite strand. I think I may be off by this, but I believe this is the case. So they can detect if there's some error that's taken place, and they can go back and replace the nucleotide with the right nucleotide. And what you do see is redundancies throughout biology. You see redundant genes, you might have redundant systems in the human body. A beautiful example is, I think it's called the laryngeal nerve. And what happens is there's this nerve that has this really long loop. And people said, oh, that shows poor design, because why would a good designer create a nerve with such a long loop? Well, when engineers looked at this, they said, this is exactly what we do. We create loops in our chords with slack for a lot of reasons, partly to piggyback other chords, partly to deal with motion. But in this nerve, what happens, you have two separate nerves going to your vocal cord from different directions. It's redundant. So if you cut one in an accident, the other still works, and vice versa. So the same way you have redundancies in human engineering, you see the same types of redundancies in biology, both in the cell and in more of the large scale anatomy, for the very same purpose, to deal with problems.
Good. Why do origin of life discussions ignore the fact that any life which spontaneously emerges has to have some way of surviving before it can reproduce?
Yeah, that's a really insightful question, because if you look at attempts to figure out what did the first organism look like, you have to assume it ate something, whether it's some sulfur compound, or you're feeding off of a thermal vent, or whatever. And the challenge is, cells are designed to feed off some food source, but the conditions necessary to produce a cell, even in one's wildest imagination, may be different from the conditions that provide that food. So if a cell spontaneously formed, it's not likely going to be in exactly the right place to eat whatever it's designed to eat. That's a huge problem. So people don't really think about that issue very much.
If we grant the miraculous, biochemical replicating hardware arises by chance, what of the software algorithm for the replacement policy and spell checker we find in cells in correcting errors? How does the cell know what an error in the genetic code actually is so it can correct it? Wouldn't that imply knowledge of what the biological code should be? Where does that special knowledge come from? Does DNA have error correction codes?
Yeah, and that's a great question, because again, what happens is DNA has all the information necessary to build your proteins and so forth. But again, it also has, I think, 3 error correction mechanisms. So well, there's 3 reasons it actually is so accurate. Part of it's a natural attraction, but part of it is these ways where biology can determine if things are out of place. So yes, there's error correction in DNA, which ensures the fidelity. And again, this question of purpose is pervasive in biology, because every component in a cell is for a purpose that engineers can often predict. So a cell has to be entirely intact, or it won't work at the most basic level. It'll break apart and turn into simple chemicals. So this issue of purpose, of irreducible complexity, of error correction are all interconnected in the design of life.
The response to Helen Hansma's theory that life could have begun between mica sheets in the ocean seems to indicate that the likelihood is infinitesimal, but does not rule out the possibility of occurring if given millions of years to occur.
Okay, so what happens is that gets into the nitty-gritties of a probability. So if you look at like Crick's fluctuation theorem, if you look at Evans-Searle fluctuation theorem, Jeremy England's work, and you start to run the numbers, the probability of simple chemicals on the early Earth turning into the simplest possible cell are numbers like 1 divided by a 10 to the power of a billion. That's like a 1 over 1 with a billion zeros. So the lowest probability that anything can happen in our universe, our visible universe, would be like the number of atoms times the number, the age of our universe times the fastest interactions can take place. Based on that, the probability of anything happening that's less than 1 over 10 to the power of 150th is essentially impossible. That's a 1 with 150 zeros. So for life to go against the thermodynamic barriers is a 1 divided by a 1 with more like a billion zeros. And you could quibble, is it a million, 100 million? The point is it's not going to happen within our universe. And even if we had a virtually limitless number of universes, the probability is essentially zero unless it truly is an infinite number of universes.
Okay, how plausible is Roger Penrose's conformal, cyclical, cosmological theory? I don't even know what that is.
This is a completely different topic, which deserves its own lesson. But what's happening is another evidence of a creator is that the universe had a beginning, that time, space, matter, and energy all began. One way around this problem is that you have a universe that's cyclical, where it expands, it contracts, it bounces, it expands, it contracts forever. So there's no beginning, so you don't need a creator. And then you've got various authors that who've tried to get around this. Like Steinhardt is an example who's promoting this cyclical idea of a bounce. The problem is that you have to create a mechanism at each bounce which resets the entropy to a very low level. And those mechanisms are incredibly contrived for that purpose. And there's absolutely zero evidence they could exist. They do exist, and they would be completely unlike anything we've ever seen. So that proposal at this point is pure speculation that has no basis to believe it.
Given our lack of insight into where life comes from, how confident can we be that there is life anywhere else in the universe?
We can be very confident that there is not life in the universe if it was produced by a solely natural process, because the thermodynamic issues don't simply relate to Earth, but anywhere. And if you want to have a self-replicating system, carbon really is the only molecule that you can use. Silicon is 2, the atoms are 2, the molecules are 2 unstable with silicon. And carbon allows the complexity of structure that no other molecule will allow without the sp3 hybridization. And if you want any self-replicating system, you're going to need lots and lots of information. So the same problems I talked about with our life, no matter what you imagine life to be, to have any system of complexity that can process energy, that contains information, you're going to need an intelligent agent to bring that about. So there might be life in other parts of the universe, but that life would have been created by God, just like we were. And C.S. Lewis postulated that possibility, but we just currently have no evidence for it at the moment.
Okay. NASA is collecting samples from an asteroid that some claim will inform us about the origin of life. Why do they say this? And is this claim true?
What happens is when you look at various asteroids or meteorite crashes, you can find certain precursors to biochemical building blocks. Like you might find some carbon-based materials, maybe something that could help produce an RNA or something. So that is what gives them hope. They think maybe these meteorites, asteroids, whatever, could have provided the chemicals needed for the origin of life. The problem is they're always, again, in an intractable mixture with lots of other stuff. So you don't really get around the problems we talked about because you still have to extract it. You've got to purify it. You've got to then have information to put all the pieces together. So it's not really that helpful for origin of life.
Okay, suppose NASA eventually sends astronauts to the moon, Titan, to the moon Titan, and when landing there, they encounter what looks like a computer and a processor, memory storage, and software code. What would the astronauts reasonably conclude as to its origin? One of our fallen satellites, perhaps? Chinese or Russian astronauts beat NASA there? Or aliens beat us there? Wouldn't each of these assume an underlying intelligence? Or was it rocks and erosion that did it?
Yeah, and that's a great idea of the design inference, because you'll hear people say it's unscientific to conclude design in nature. But again, if you found a computer mechanism on a moon of Jupiter, or my favorite example is the movie Contact. So in the movie Contact, there's a signal from space, and then Jodie Foster realizes that this signal has a series of prime numbers, which you're not going to get from any natural process. And there's the encoded instructions for a spaceship. Now, if you use the logic of scientific materialism and you don't believe in aliens, you'd say, well, it's unscientific to argue that you see design from information. It's much more scientific to argue that there were self-replicating quasars or self-replicating stars that were being selected for spaceship emitting signals. It's sort of the same logic. So when you look at it, the simplest possible cell, you see information with instructions, and you see machinery, you see energy production, information processing, error correction. So in the same way, you know that your artifact on the moon of Jupiter or the signal from space in contact is designed, you know that life is designed also.
Okay, some Christians with reservations about the science of evolution separate those from issues of teleology, which they believe belong to metaphysics. Do you agree that these are separable?
No, I don't. Because if you look at, let's say, and again, there's a lot of very, very sincere Christians that want to promote evolution in the church for very noble reasons. They want to remove conflict. Very nice people, really respect their hearts, and I appreciate what they're trying to do. The challenge, though, and this is what people don't realize, is that if you look at Darwin's personal writings, when he spoke publicly, he would use God talk, but privately, he acknowledges that he was a materialist. He embraced this idea there was no creator, everything was through natural processes. And when he developed his evolutionary theory, he actually read William Paley, and he used the same arguments of Paley, who had the famous watch argument, the same design language, the same examples, but he replaced the creator, God, with natural selection. So natural selection became a demigod that removed the need to believe design in biology. And it's the exact same thing with the ancient Greeks. If you look at the people like the Epicureans, who were influenced by the atomists, by Democritus, you look at like Lucretius, and what they did was they said, we see design in life, but we don't believe the gods were involved in the world, so we'll promote an idea of natural selection to remove the need for design. And when Paul in Romans 1 was talking about people suppressing the truth, who were ignoring the evidence of God's power in creation, the group he was engaging were the Epicureans, who believed evolution replaced the need to believe in a creator for the appearance of design in life. So evolutionary biology, as it's taught in most universities and high schools, presupposes that everything is a product of blind, undirected processes. There's no design. It's not a secondary cause used by God. And that's why they assume things are going to be suboptimal. They'll assume things are poorly designed. That's why in the 80s, people believed that a large portion of the human genome was junk DNA. Like 90-some% they believe was junk DNA, because they assumed that had to be the case because it was naturally produced. And then when people realize it's not junk DNA, but most of it we can tell is functional, a lot of evolutionary biologists said if ENCODE is right, if that much is functional, evolution is false. So again, at a very deep level in terms of how the science is done, it's anti-teleological. So the philosophy and the science are integrated at a very, very deep level. Also, as you look at systems biology, to make progress, they use the language of teleology. They assume purpose. So the philosophy of teleology and practical everyday research in biology are intertwined. They cannot be separated.
Okay, are scientists aware of any natural process that gives rise to biological information?
Great question. And this really deserves its, this is a long topic, but the answer is no. And I have a whole series of articles. If you go to look up Brian Miller, Evolution News, look up my articles. I have a whole series on the information enigma dealing with that claim that natural processes can create information for free. I'll just quickly give an analogy. Imagine that you are sick and you come downstairs and your mother prepared you a bowl of alphabet soup, that soup with letters in it. And you notice in that soup that you have the sentence, I hope you're feeling better, get a lot of rest, drink a lot of fluids, take care of yourself. Could you explain that message in the soup by the chemistry of the pasta? or the physics of the boiling water? No, because whenever you have the arrangement of letters for a purpose, that always points to design. Now, you can create small amounts of information by chance. Like if you saw the word cat in your soup, you might assume that was by chance because that's not that improbable. But a very long sentence you're not going to see is there by chance. In the same way, if you look at the research on proteins, we have Doug Axe's research on proteins, and I continued that research by drawing upon the research of Dan Tofik. And based on his research, a direct mathematical analysis on his research, the probability of a random sequence forming a functional protein is 1/3 to L, where L is the length of the protein. So the largest protein that could be produced by chance would be less than 100 amino acids. Most of the essential proteins in life are much larger than that. So we know that there's no natural process that's going to produce large amounts of information that could help with the origin of life. In fact, you couldn't even produce a simple protein through a natural process because the probability of hitting that target is too small. And you have what are called no free lunch theorems, which show you that no strategy for finding a target on average is any better than a random search. So there is no natural process that will create information at a rate that's more efficient than a random search. And the amount of information you can create in a random search is very, very small. So that's just sort of a quick answer to that.
In the table comparing computers to cells, I didn't see a RAM chip in the cell column. Is it because we haven't discovered it in cells, or because RAM, which is random access memory hardware, isn't absolutely necessary for cells or computers to operate?
Right, and that's a great point, is you have to recognize that biology is different from human engineering. That's obvious. And it's different in the ways it's better, it's more efficient, it uses principles we probably don't even understand. So you're not going to see necessarily a perfect one-to-one correspondence in terms of the parts, but you will often see a beautiful correspondence in terms of the function. And I thought in that research paper, they actually did mention RAM. Like they talked about things like the the messenger RNA, different things like that that may function like RAM. But you'd have to go through the paper to see the exact details. So don't expect there be an exact correspondence of parts, but expect the functions to correspond very closely in many, many cases. So that's just the key difference to make.
Okay, last question. Where does design fit in Genesis?
Oh, I mean, that's a great question. And obviously, different Christians will debate certain aspect of Genesis. Like the more traditional young earth creationists would argue that that each day represents a design event. Day one, God created sun, moon, and stars, and so forth. Someone who's, let's say, an old earth creationist would argue that you see sudden points of creation, but maybe over longer periods of time. Other people might argue maybe it was more of a gradual process, but God infused the information more gradually as things are forming. So that gets more into the theology and the biblical interpretation, which is not my expertise. So that's probably good for another program. But let me just talk about the science. When you look at the fossil record, what you see is sudden appearances of radical new organisms without precursors. The classic example is the Cambrian explosion, where you see the first representatives of a lot of the major animal types appear suddenly without clear ancestors going back to the trunk of a tree. So, and there's many examples of like that in the fossil record. And the time period is on the order of like 1 to 10 million years when the largest transformations take place in nature. And if you look at papers like Dredd and Schmidt, you look at other analyses of how long it takes for new information to enter into the genome and spread through the population. For instance, like in fruit flies, it would take about 9 million years based on Dredd and Schmidt, their 2008 paper, I believe, for 2 specific coordinated mutations to appear and spread through the population. So in the time in the fossil record that you see the largest transformations, it's only enough time for 2 specific mutations to appear and spread through the population. In humans, the time it would take for 2 specific mutations just to appear would be over 100 million years. Well, the hypothetical lines going back to taking us from humans and chimpanzees to a common ancestor would be more like 7 million years. So the time scales indicate that there's massive infusions of information throughout the geological column where new life appears, and that's clearly where you have design.
Great, great. Well, Dr. Miller, Dr. Brian Miller, thank you. Thank you for this, and thank you for sharing. And you are a world's authority in the thermodynamics on the origin of life. And so I appreciate that we had the opportunity to get the very, very best. God bless you, my friend.
Thank you.
All right.
Thank you. I greatly appreciate the time here.
Thank you, and thank you to our guests and our virtual questioners, and thank you for joining us at this Science and Faith podcast. God bless.